Hello everyone, welcome to Biochemistry Simplified. We are discussing medical biotechnology, and in medical biotechnology we've been discussing chromatography. Today we are going to discuss a very important technique of chromatography, that is hydrophobic interaction chromatography. In this technique we have a column containing the material with hydrophobic groups, and these hydrophobic groups can be methyl group, ethyl group, propyl group, octyl group or phenyl group. These are the different hydrophobic groups present in the material of the column. So column is basically hydrophobic in nature. At high salt concentration, all the proteins are hydrophobic. Very important point. At high salt concentration, hydrophobic. High salt, hydrophobic. At high salt concentration, all the proteins are hydrophobic. Some are more and some are less, but all are hydrophobic. So all the proteins will bind to the hydrophobic groups, to the stationary phase, but some will bind tightly and some will bind loosely because their hydrophobicity is different, right? So mixture of proteins at high salt concentration, all the proteins are hydrophobic. Some are more, some are less. That is a different matter, but all are hydrophobic. So this mixture of protein is poured over the column. So all the proteins will bind, but they will bind with different strength. So all the proteins are bound because all of them are hydrophobic. Now how these proteins are removed? These proteins are removed from the binding sites by changing the salt concentration. If the salt concentration is decreased gradually, the first protein to be removed will be the one which has least hydrophobicity. So proteins are eluted sequentially in the order of increasing hydrophobicity. So first protein will be the least hydrophobic, then more hydrophobic, and then the most hydrophobic protein, because salt concentration is decreased gradually. So that is one way of eluting the proteins on the basis of their hydrophobicity. Apart from salt concentration, we can also use some other elutant like increasing the concentration of the detergent, because detergent will break the hydrophobic interactions. Detergent will disrupt the hydrophobic interaction, very weak forces. So initially small concentration of detergent, so least hydrophobic will be removed. Then more detergent concentration, more hydrophobic will remove. Then increasing the detergent concentration further, then the most hydrophobic protein will be removed from the binding site and eluted. Apart from detergent concentration, we can also do the same thing by changing the pH. But generally we use the salt concentration or the detergent concentration in the order of increasing magnitude. Advantages of hydrophobic interaction chromatography is, if the sample is not sensitive to high salt concentration but sensitive to pH. See, proteins are very sensitive to various physical conditions like temperature, salt concentration, pH. So the protein has tendency to get denatured if the pH is too high or too low, salt concentration is too high or too low. So if we have the proteins which are not sensitive to high salt concentrations but sensitive to pH, so we use the technique of hydrophobic interaction chromatography. Or if the proteins are sensitive to harsh solvents, because in the other techniques of chromatography, harsh solvents are used. So there also this hydrophobic interaction chromatography is useful. So if the proteins are sensitive to pH and salt concentration both, at fixed pH and salt concentration. See, earlier we were using high salt concentration where all the proteins were hydrophobic and they will be eluted one by one by gradually decreasing the salt concentration. That was one way of doing the hydrophobic interaction chromatography. Now I am going to tell you another way. Proteins are sensitive to pH and salt concentration both. But when the pH and salt concentration is fixed, some of the proteins are hydrophilic and some are hydrophobic. So those which are hydrophobic, they will bind to the stationary phase. Hydrophilic will not bind. Now we have a mixture of proteins at a fixed pH and salt concentration. Some are hydrophilic, some are hydrophobic. So when this mixture of proteins is poured over the column, the hydrophobic proteins will bind because the material of the column is hydrophobic. But hydrophilic proteins will be collected at the bottom of the column. Now the hydrophobic protein has to be removed. How? It is removed by adding another molecule with more hydrophobicity, and a very good example in this case is isolation of immunoglobulin G from the serum. In the first step, in the first stage, the serum is added. The immunoglobulin G will be separated from rest of the proteins. It is bound to the material of the column, and now it is removed by a molecule with more hydrophobicity, that is beta cyclodextrin as a competitor. Very important molecule. Hydrophobic interaction chromatography, one very specific use is isolation of immunoglobulin G using beta cyclodextrin as the competitor. Okay, so beta cyclodextrin will simply remove the immunoglobulin G from the binding sites, and that will be collected. So the points to remember about hydrophobic interaction chromatography: at high salt concentration, all proteins are hydrophobic, and at specific salt concentration, some proteins are hydrophobic, some proteins are hydrophilic. And hydrophobic interaction chromatography is also called reversed phase liquid chromatography, RPLC, because in the normal chromatography, mobile phase is non-polar, stationary phase is polar. But in hydrophobic interaction chromatography, the stationary phase is non-polar, mobile phase is polar. So that is why it is called reversed phase liquid chromatography, RPLC in short. And by eluting, when all the proteins are hydrophobic in the first instance, all the proteins are hydrophobic, so they are eluted by changing salt concentration, and they are eluted in the order of increasing hydrophobicity. And one very specific example, when there is salt concentration is specific, then immunoglobulin G eluted with beta cyclodextrin as the competitor. So these are the points that you must remember. So that was hydrophobic interaction chromatography. Let us now discuss another technique of chromatography, that is called size exclusion chromatography. In size exclusion chromatography, we have a column filled with porous beads, and porous beads of polyacrylamide, dextran or agarose. Polyacrylamide is commercially known as Biogel P. So P for poly and P for Biogel P, or cefacryl. So Biogel P or cefacryl is a name, commercial name given to polyacrylamide beads with pores. Similarly, dextran is known as cefadex, agarose is known as cefarose. So these are the porous beads. These porous beads, as you know, proteins have different sizes. The smaller protein can easily enter into the beads, the larger protein cannot. So when there is a mixture of proteins with different sizes, and this mixture is poured over this column filled with the porous beads, those proteins which are larger in size will come from the sides of the beads, so they will be eluted earlier. This is important. Larger proteins will be eluted first. Smaller protein will enter into the beads and will come slowly, will be retarded. So smaller proteins will come later. So larger protein will come earlier, smaller protein will come later. That is called size exclusion chromatography, which separates the proteins purely on the basis of size. To be more specific, proteins are separated purely on the basis of tumbling radius or Stokes radius. So tumbling radius and Stokes radius is a more specific answer, and you can simply say it is size, but more specifically it is tumbling radius or Stokes radius. This technique is also called gel permeation chromatography, gel filtration chromatography, or molecular sieving. So these three names are given to the same technique of chromatography. So don't get confused. So remember, size exclusion chromatography, gel permeation chromatography, gel filtration chromatography, and molecular sieving. These are the names given to the same technique of chromatography, which separates the proteins purely on the basis of tumbling radius or Stokes radius, right? So that was size exclusion chromatography. So We have discussed the various techniques of chromatography. We have discussed paper chromatography, we have discussed thin layer chromatography, gas liquid chromatography, affinity chromatography, ion exchange chromatography, hydrophobic interaction chromatography, size exclusion chromatography. And a few points I have already told you about each chromatography. Let us now revise the important points to remember about the whole discussion of chromatography. In paper chromatography, amino acids are separated on the basis of polarity. Non-polar is fastest. This is important. Non-polar, more fast. Okay. Amino acids are separated on the basis of polarity, and the most non-polar will be the fastest. TLC, use of TLC for drugs use and abuse. Drugs of use and abuse. And same is with GLC, lipids, steroids and drugs, right? RF value. RF value stands for retardation factor. To memorize, you can memorize it as ratio fronts, RF. Distance traveled by individual amino acid upon the distance traveled by the solvent front. It is different from the retention factor, which is used in column chromatography. That is the time analyte retained in the stationary phase upon the time analyte is retained in the mobile phase. These two are different entities. Okay, RF value and the retention factor are different. Materials used in TLC, SAC, materials used in column, CDAPs, GLC is used for lipids, steroids and drugs of use and abuse. Detector and recorder system to remember spectrophoton. And the most specific chromatography for proteins is affinity chromatography. It is used to purify the proteins, and the name of ligands is very important. Which ligand for which protein, that we have discussed in the previous lecture. So these are the points to remember. Some more points to remember. The technique separating proteins purely by size, gel filtration, gel permeation, molecular sieving or size exclusion chromatography. And to be more specific, these all mean the same. All are the names given to the same technique, and it uses Cefatex, Cephorose or Cefacryl or Biogel P columns, Dextran, Agarose or polyacrylamide, and it separates the proteins by tumbling radius, Stokes radius. These are the points about the size exclusion chromatography, and the proteins larger than pore size are eluted earlier and smaller ones are eluted later. Techniques separating proteins purely by charge, ion exchange chromatography, and it is used for HbA1c and other hemoglobin variants. Hydrophobic interaction chromatography, also called reverse phase liquid chromatography, and the immunoglobulin G eluted with beta cyclodextrin as competitor. Chromatography with best resolution is HPLC. Now what is HPLC? HPLC. HPLC. HPLC is a technique which uses small adsorbent particles with the average size of 2 to 50 micrometer. The particles are very small. The material filled in the column is small size particles. It is the technique in which the sample is forced along with the mobile phase with high pressure. For forcing the sample, we use high pressure from one side, and because high pressure is used, it is called high pressure liquid chromatography. HPLC stands for high pressure liquid chromatography. But what is the advantage of using the pressure and using the small sized adsorbent particles? The advantage is to have the best resolution. So the technique with the best resolution is HPLC, and that is why it is also called high performance or high precision liquid chromatography. Okay, so HPLC, best resolution, high pressure or high performance or high precision liquid chromatography. And because the pressure is used, the column has to be robust, strong column, that is stainless steel columns, and any type of chromatography can be done with the help of using high pressure or high precision. So that is HPLC. So these are the points to be remembered about the HPLC. Okay, so that with that we finish our discussion on chromatography. In the next lecture, we will be discussing the techniques of electrophoresis. Thank you.